Hello, students, and welcome to my screencast for your Unit 6 test on mitosis and meiosis. This first part will be on cell size, methods of reproduction, cell cycle, and mitosis. This is in your textbook, Chapter 10, Sections 1, 2, and 3. Don't sleep on the textbook. It's been a while since you've done the reading from it, but I'd encourage you to go back and look at that active reading, especially to look at the figures there. Remember, if you can explain it to a family member, a friend, anybody, then you really understand it. All right, so really try and explain this material and act it out and make models and engage in the scientific practices. All right, let's go ahead and jump in. These are some pictures from this weekend. A lot of fun thinking about mitosis. Here we go. Oh, just real quick, I'm going to use a lot of Khan Academy images. They're available for free. The content is on khanacademy.org, and I have licensed this that I will not make any money off of this, and nobody else can as well. It's meant for that to help you learn. Here we go. Okay, cell size. This will not be on the test. So if you want to fast forward, you can, but it was in chapter 10, and it's just kind of a reminder that cells have a certain size and why? Why are they here? Why aren't, cells as big? Why aren't we just one big cell? Or why aren't they even smaller? Can you answer that? Well, maybe you remember that cells are trying to maximize their surface area to volume ratio. That's kind of a tough tongue twister, a convoluted way of saying they're limited in size. If they get too large, they're going to have trouble exchanging materials, food, oxygen, water, and they'll overtax their DNA. But if they're too small, then they can't perform their specific functions. So they're trying to find a sweet spot. And that sweet spot's governed by a little bit of math with surface area to volume ratio. But that was on a couple tests ago. All right, so here we go. This is one place we'll start that will be on your test. And so methods of reproduction, you have asexual reproduction, just like mitosis is, genetically identical offspring. Can you think of, without just reading the slide, sorry, I should have animated it, what are some pros and cons of asexual reproduction? Maybe you can tell a friend. All right, as you can see here, the pros is be very fast, and the cons would be that it lacks genetic diversity. So if you're in a very stable environment, maybe asexual reproduction is really good. And we can see that in bacteria. We can see that in, we watched that TED Ed. It dealt with even some snakes that can do it, and even turkeys. All right, here's an example of binary fission in bacteria. So it's just going to copy its DNA, clone itself, and split. Now, it's a little misleading, right? There could be mutations that happen within there, which is one of the reasons we've led to bacterial superbugs, but we'll get to that in our evolution unit. Here's an example of budding. So this hydra is just budding off and making a new one. Can happen in potatoes too. Another example that I don't have a picture of in here would be sporulation, like fungi sending spores out that can become more fungus somewhere else, but they'll be clones. The coral that you see are all clones of each other as they become one colonial organism. If you're out there diving into the minions here in Omaha. All right, how about sexual reproduction? Ooh, sorry, should have animated that. So, can you think of some pros and cons of that? Well, hopefully, if you didn't just read off the slide, some pros would be it's going to make genetic diversity, right? So, in the face of a changing environment, to have genetic diversity in a population is very important, so that way the population can survive and go on. The cons is that it takes time, right? It's a lot easier to just reproduce asexually and just make a clone of oneself. It takes time to find mates, and it requires energy. Okay, oh, my head is covering this up. I'm not able to move my head, so you're just gonna have to see through it. All right, so here would be the human life cycle. And so males and females make eggs and sperm, respectively. And those eggs and sperm will be haploid. They will be one in, or we just said in class, in. Here the example is 3. So each sperm would have 3 chromosomes. They're the sperm ones. Each egg would have 3. When they combine together, they make 2 in is equal to 6. So this is called a diploid cell. This is the zygote, the very first cell of what we all started as. The transcription factors will be on different sides of this zygote. When it's asymmetrical, when it divides, it'll start to specialize, and it'll go through and make the wonder that is you. So let's repeat, right? Basically, we undergo meiosis to make sex cells. The sex cells are a haploid. You can remember haploid has half the number of chromosomes, students. And then when they come together, they make diploid cells. This is a zygote. And then they're going to reproduce many times through mitosis, trillions of times, in fact, to make you. So ask yourself, what are the haploid and diploid numbers for humans? That's just something you should know, you should memorize. Hopefully, you got the haploids numbers. N is equal to 23. So dad's going to carry 23 chromosomes, mom's going to carry 23 chromosomes, and you are going to have 46. Then, as you want to continue your family line, you'll make eggs or sperm that have 23, and your future mate will have the same. Okay, so that was pretty easy. Hopefully, a good little quick review. So let's talk real quick about the cell cycle and mitosis. We'll deal with prokaryotes first, or bacteria, and then we'll get into eukaryotes. All right, let's dive in. So, do you remember how bacteria reproduce? We did this very quickly in class. I should have spent more time on it. But bacteria just have a single chromosome, circular. They can just copy it and then divide. All right, usually that chromosome kind of is attached to the cell membrane. And here's a picture of it. Sometimes you can insert what's called plasmids, or bacteria will have them as well, just naturally. Plasmids are little circular DNAs in here, and that's actually how we make insulin, right? We put a plasmid in here that codes for, it has the genes for making insulin. But that's an aside. So what do you need to know? Binary, it means it splits in two. Fission, so the fission is the splitting. It's just going to copy the DNA and split. Pretty easy. Well, we evolved from bacteria, so you'll see some similarities. You'll see that here in mitosis. But before we jump into that, let's talk about what are chromosomes, right? We're about to get into this, and I find just from years past that students might struggle a little bit with exactly what is a chromosome. So ask yourself. Okay, hopefully you got something along the lines of chromosomes are made up of DNA and genes. I'm not worried if you know these words: chromatid, histones, nucleosomes. That's all kind of AP stuff. But what you should know is that DNA is in the nucleus. DNA contains genes. A gene is a segment of DNA that codes for something. Usually, it codes for a protein. So this segment right here might code for the protein for part of your hair. And it might be whether or not you have a widow's peak, or whether or not you have a straight hairline, whether or not you have attached earlobes or unattached earlobes, what your blood type is, whether you're I am B positive. So whatever you are. All right, so that is a chromosome. This is called a chromatid. I believe that's on the next slide, and they're going to be identical copies of each other. These letters are the bases in DNA, and that will be in our upcoming unit. We'll learn how to read those letters and turn them into mRNA transcripts and into proteins. So here's a picture. I said a couple times in class that the DNA resembles spaghetti in there. Now, hopefully, you can see it. So it's called chromatin. It's not just DNA. There's proteins in there as well. So it's going to copy itself using an enzyme called DNA polymerase. It's going to copy the DNA. And once it does that, they can form chromosomes in mitosis. And here's the chromosome. The centromere is that middle button. And this is a chromatid. These are identical copies of each other. All right. Here is a human karyotype. So tomorrow in class, you'll get to make one kind of online quickly. It'll be fun. Notice how they're about the same size if you kind of stretch them out. That's how we order them. So 1 would be the biggest, and then 2, then 3, all the way down to 22. And then we keep the sex chromosomes separate. All right. You can see that 1 is from mother and 1 is from your father. This is what's called a homologous pair of chromosomes. Homo meaning the same. They have the same genes, but they may have different forms or alleles of those genes. Alrighty then. See if you can remember some of this terminology here. So pause for a second. Okay, here it goes. Haploid means one set of chromosomes or in. Gametes are sex cells. Diploid are two sets of chromosomes or two in. And somatic is body cell. I needed to have used this word more in class, and I will. So mitosis deals with diploid somatic cells. Meiosis uses diploid germline cells to make haploid gametes. There we go. All right, I'm sorry my head's covering up again. This is just some helpful terminology. Here's another picture of a chromosome. So it really confused people in class counting chromosomes and chromatids. I really encourage you to check out that Amoeba Sisters video on it. I'm not going to play it in class. Unfortunately, we just don't have time, but it's something that you can learn from on your own. Here's my trick is to always count the centromeres. That'll tell you how many chromosomes you have. If there is no centromere and they're in separate cells, then a chromatid can count as its own chromosome. So let's look at this picture. This is from Khan Academy. Here's one chromosome or one chromatid. It is now being copied, so there's going to be two of them. It's in this form, right, that it could be read in a cell and that genes could be accessed and it can make proteins. Maybe it can make something like catalase that you would need. Here they have condensed to undergo mitosis, so this would be in prophase. And then they separate in anaphase. And so once they separate, they become their own chromosomes. That's just how we count them. Alrighty then. Check out that video if you're confused. This right here should say 2 chromosomes, 1 chromatid underneath it. Okay, do you remember the stages of the cell cycle? You go ahead and take a guess while I take a sip of water. Thank you. All right, we have G1, S, G2, mitosis, which is composed of prophase, metaphase, anaphase, and telophase, and cytokinesis. G1, S, and G2 make up what's called interphase. Notice how interphase is much longer than mitosis and cytokinesis. During interphase, the cell does the work of its life. It does the work that it's needed to keep you alive. And when it needs to divide, then it goes through the process to make more of them. And it's going to separate during cytokinesis. Mitosis is division of the cell nucleus. This kind of gives it away that G1 is when the cell is growing. S, it copies the DNA. G2 continues to grow and it prepares to divide, and then it goes through it. Here's my little schematic. My head's covering it up again, but I have 1, 2, 3, 4 chromosomes here. So they're going to be copied, and you're going to have identical daughter cells down here. Notice how the daughter cells are a little smaller than the parent cell. They will have to grow during G1 to regain that size. All right. Can you do it with your hands? Do you remember? Well, here we go, just in case you can't. So we have G1. Here's my cell growing. You could say it grows. S, it copies its DNA. G2, it gets ready to divide. Prophase, it starts to form chromosomes. Metaphase, they all line up in the middle. Anaphase, all the chromosomes come apart. Telophase, T for 2. And then you guessed it, our favorite, cytokinesis. They chop it in half. So let's see some pictures of it. We're going to roll through this pretty quick. So here we have prophase. The chromosomes start to condense. The nucleus is gone. That's the area that makes ribosomes. The mitotic spindles start to form, and then by late prophase, the nuclear envelope has broken down. The chromosomes are fully condensed, and we're ready to get the move on. So P for prep. Here we go. Chromosomes come together. Can you identify the homologous pairs of chromosomes here? We have 2n is equal to 4. So this would be the homologous pair, and this would be a homologous pair. You could say this is from dad, and this is from mom, dad, mom. Okay, so I'm not going to make it, I didn't ask you to memorize any of these terms, etc. But I did just want to show you, here's how the spindle fibers attach right there to the centromere. they're going to attach, and another one's going to attach to this side, and that's how it pulls apart those chromatids, right? In meiosis, it's going to grab onto the whole chromosome. Here is metaphase. They all line up in the equatorial plate, or right there at the equator, right there in the middle. Bang, they get pulled apart. So a good question to you is, did it get pulled apart, or do motor proteins walk them there? It's for you to figure out. And then remember, A for away, anaphase. Here's telophase. We got two separate ones. All right, we have the spindle fibers start to disappear, the nuclear membrane reforms, the chromosomes start to decondense, and you have two new nuclei, and then the cell will split. Animal cells, it splits with a cleavage furrow. In plant cells, it's going to form a new cell wall or a cell plate right here. Quick reminder, the cells can exit the cell cycle. They can exit at G1 and go to what's called gap zero, a resting stage. You might have heard that your brain cells do that. All right, can you find some stuff in your house and model mitosis? Right, you're allowed to check out the pop beads with me if you want. We unfortunately didn't do Play-Doh this year. I do have some pipe cleaners, but all my Play-Doh got messed up by my kids. So here we go. Now we're going to copy that, replicate that DNA. So we got our two chromatids here. This would be 2n is equal to 2. Centrioles, they get pulled apart, and you got your new cells. All right. Woo, this slide is a handful, right? I'm not going to sit here and read it to you. But the last part that we're going to deal with is cancer. So this is really important to me. I actually picked, I'm doing this screencast in my house, and you can see behind me, that is my mother. She was in college. She was a doctor and got me interested in science. Okay, so let's go through. We'll kind of hit the information here by talking about it. What happens at a checkpoint? If you're going to get stopped at a checkpoint on a road, what are the police looking for? So hopefully you got something along the lines of that you had the proper registration, that you were going the right speed limit, that you're of sound mind and body to be driving. Well, the cell is going to do the same thing. So can you guess what's going to happen during the G1 checkpoint right here? If you guess that it's checking that the cell is pretty much, it's just checking that it's ready to divide, that it's the right size, that it's got enough food and nutrients to do it, that the appropriate growth factors are telling it to divide, and it's assessing whether or not it has any damage to its DNA. Right? So is it large enough to divide? Can we do this? And is any of the DNA damaged? If anything's wrong, the cell can try and fix itself, or it can program itself to self-destruct. And that's called apoptosis. That's a good thing if it self-destructs, if it was going to have a mutation that could have caused cancer. Can you guess what happens during the G2 checkpoint? Right? Think about what's different between G1 and G2. Hopefully you see that we've gone through this S phase. So if you guess, it's really checking that the DNA was replicated properly. That means that it was copied properly, and it's also checking for any damage. Now can you guess what happens at this mitosis checkpoint? Right, just be right around metaphase. Well, I kind of gave it away, right? It's checking that the chromosomes are attached to the spindle properly, and that everything is ready to be divided, so that you get the right number of chromatids, which will then be labeled as chromosomes in each cell. All right, checkpoints sound kind of simple. Could it be a little more complicated than that? Yes. So we did not get to talk about this enough. You did the Quick and Learn from HHMI. We watched a couple of video clips on it, but I'm not at the point where I feel comfortable testing you on this, so you can fast forward if you want. But what really happens is that you're making proteins called cyclins. The cyclins, as they rise in number, will activate these cyclin-dependent kinases. These will phosphorylate other items in the cell. That means they attach a phosphate to it, and it's going to cause action to happen. So as enough M cyclins come around, it's going to kick off these CDKs, which here is going to break down the nuclear membrane. And there's different types of cyclins that cause different actions in the cell. So all these are ways that the cell cycle can be controlled or that we can lose control. Look at this. This is called a karyotype. I said earlier you get to make it in class. Here you see 1 is 23. Is it a boy or a girl? Hopefully you got that it's a girl. 2 X's there. And here you can see a cancer karyotype. And just notice the differences. We have a lot of chromosomal mutations and extra copies, etc., that have happened in this cancerous cell. It is flying through those checkpoints. It is not stopping for them. Something is wrong. The cyclins could be out of control, etc. All right, let me stop flipping around. Sorry about that. Here are 3 probable causes of cancer. This slide is actually from a college lecture that I went to about cancer. And so it could be a defective DNA repair mechanism. We didn't get into that too much. We'll do that more with our DNA unit. It can be the transformation of a normal proto-oncogene into an oncogene. Onco sounds like oncologist, like my mom out there. So dealing with the genes of cancer. So when a proto-oncogene gets mutated, it becomes an oncogene, and what that means is it becomes a gas pedal, and that gas pedal is stuck downwards, saying keep on dividing cell. Or it could be a malfunction or mutation of a tumor suppressor gene, like P53, which means that you lose the brakes. So if you're in a car, you would have no more brakes to stop cell division if something is wrong. So let's check out a couple of examples. Here's just a nice little graphic. Once again, my head's in the way. Get your healthy cell, some type of damage happens. It could be an environmental damage. It could be smoking or vaping or radiation, or it could just be something internally goes wrong. Your cell will hopefully check itself at one of those checkpoints and decide to either undergo repair and it can successfully return to the cell cycle, or if it stops at repair, it can become cancer, or if it has too much damage, the cell could just die, right? And you would hope that it would program itself to die rather than become cancerous. Here is an image from our HHMI Quick and Learn. I hope you enjoyed that. I really enjoyed seeing you engage in that science. And so here are some tumor suppressor genes. And these tumor suppressor genes, if they get mutated, mean that you no longer have the brakes on the cell cycle. And here could be some proto-oncogenes. So if they get mutated, you're stuck with the gas pedal on. Here's a cool AP example of it, just showing this is called well, you know this, right? This is a ligand, and this is a signal transduction pathway for a cell to divide. And what happens if it gets mutated? It gets mutated, and it's always on. It's always saying divide, 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 divide cell, and it's going to cause cancer. Here is a simplified version of how P53, this is often called the guardian of the genome gene. Right? And I asked you about that in your Terry Fox cancer poster. So it can, it's activated by DNA damage. It can cause the production of an inhibitor for the cyclin-dependent kinase. So it stops the cell from dividing if something is wrong. So if it gets mutated, then the cell can keep on going. It has no brakes. It has no way to stop the car from going. Okay, here are my kids. You can see the smiles on their faces for their 3rd birthday. They're smiling at my mom there in the background. I wish she was still with us. And what I would just like to say about it is that, we know a lot about cancer. I hope that some of you all might become cancer doctors to help to fix it, right? We know a lot, but we still can't fix it. It's why we have all these Terry Fox runs and items like that. And then some of you that might not want to become doctors, I encourage you to get involved politically, right? Read this quote. While much is known about the science of cancer, its prevention depends largely, if not exclusively, on political action. Sorry, my head covers it up again. That's from a doctor, I believe it's the University of Chicago. I'll double check it. But there's a lot of action going on, right? Like what kind of environmental laws are we passing, et cetera, that can help to make our world a safer place and less full of cancer. Okay, students, I hope that helps you for your test. Good luck, take care, and have a good one.